and actually make the chromatin more or less tagged and more or less compact to some extent. Okay? So now you realize that along the DNA you will have some markers, for example, DNA methylation, but along the in histones as well, you will have another series of modification that will actually give clues along the genome that maybe this is to be compacted there, and this is to be placed in an A compartment, or maybe this is to be placed in a B compartment, not to be expressed. I hope I was clear with you. I'm sorry to be so slow, but it's very basic and really critical information. So now I hope that I made you understand that you can't think of a genome as a naked thing. You have to think of the genome as something which has additional layers of information that help the cell make the genome express itself. And this expression is really guided by the organization in 3D of the sequence information, the genetic information. Okay? So what we're going to do now is to tell one question, very simple. Why does that all matter? It does matter a lot because it's the way genes are expressed, isn't it? So we wouldn't be alive if that was not real. So that's very important. But it's also important for you to realize, and I had a very good question from one of you yesterday, which came to me and asked me, okay, but how do we tell whether it's DNA, so genetics, or environment that guides the traits that an animal develops? And I said, we will cover that tomorrow, and that's the time now. So I'm just showing you those two mice, and if you see those two mice, or those 6 mice, you may think they have different genomes, right? They will have somehow, maybe that one will have a mutation that causes a sort of brown color, whereas that one will have a different mutation or no mutation at all, and it will be yellow. Well, if I told you, and that's the same for those ones, by the way, right? There is an increase in weight as those ones age. As they age, they become obese, those mice. Well, you will be surprised maybe to hear that those mice are actually isogenic. It means they have the very same genome. They are clones of themselves. So that tells you that if they are clones of themselves, and they develop such different traits, that's shocking. It means that it cannot be genetics what explains their difference, isn't it? And there are those mice, plus they are grown in almost identical environments. So what is the driver of that thing? There is something that is not encoded in the genome, it's not sequence information that can drive those phenotypic drastic differences. And so long story short is due to, in the genome, there is this IAP, which is an inverted repeat that is inserted somewhere in the genome. And this is something that is a sort of LTR, which is an endogenous retrovirus, which is integrated in the genome. It doesn't matter, I don't want to go into details, but what is important is that the DNA methylation of that thing in the genome is actually responsible for the traits that develop. So you have mice that are grown in normal condition, depending on the level of methylation, DNA methylation of this retro LTR that I just mentioned, the IAP thing. If it's not methylated, this gene AVI will be expressed and the mice will be yellow. But if it's like hypermethylated, when there is a lot of DNA methylation, as one of the signals that I mentioned before, then the gene will be expressed in a way that is causing this sort of agouti phenotype, the brown phenotype. So what you learn here is that depending on the DNA methylation, the mice will express the genes in different tissues, or will express the gene differently, resulting in a yellow versus a brown phenotype. So in normal condition, what I tell you is that depending on the level of DNA methylation, and only there is no sequence change here, it's just DNA methylation, different phenotypes. So if we are interested in animal domestication, traits are important because maybe you want to breed a trait. And imagine you are a breeder, you want to develop some yellow mice. Oh, you're happy you have yellow mice, and all of a sudden they could become brown. Well, it could be a problem, right? If it's a cattle with like nice meat, and now the meat is not as good, right? So it means we have really to pay attention to those extra layers of information. Not all the phenotypically relevant modifications lie in the DNA sequence variation. They could lie in other types of variation, such as DNA methylation in my example. And so what is important here is that depending on the diet of that mice, for example, if you give the mice a diet that is rich in bisphenol A, for example, it's a diet that will actually be poor in methyl donors. Methyl is CH3 in chemistry, so the diet will have poor content of CH3, carbon and free hydrogen, methyl. So it means that DNA methylation cannot be high because the diet is deprived in the source of methylation. It will on average be yellow because you can't really methylate around the DNA. But if you do the other thing, which is you give a diet that is rich in methyl donors, such as the folate, which is one element of the diet, then because folate contains a lot of methyl group, on average those mice, they will actually have a higher DNA methylation level, therefore they will on average be more brown. So what I tell you is that even though they all have the same genome, depending on the environment they choose, the diets they eat, in fact they have, they will develop different phenotypes. So I hope that I convince you here that all those layers of information that I'm mentioning, which are a pain because it's complex to understand and it's complex to study, are yet very critical to account for when we think about phenotypic traits development, okay? And this is what I just said. Okay, so how about the methodology? So how do we study DNA methylation to start with? So there are tons of methods. I don't want to cover that too much in the details. I put that in my slides for those of you who are very interested in the methodology. But there are many different ways, but one is DNA bisulfite sequencing. You may have heard about this, which is the gold standard for having high resolution DNA methylation map. How do we know that this part of the genome is methylated? It's because we have methods that can help us tell this is methylated. And so one of the assays that people are doing to actually know where the genome is methylated is to treat the DNA with bisulfites, and bisulfite is a chemical product that actually transforms the cytosines, the Cs that are not methylated, into uracil. Right? C is transformed chemically into U, okay? Whereas if the DNA is methylated, methyl Cs remain Cs. So this is a very important thing. It tells you that we have a chemical treatment that can make cytosines into uracils if it's not methylated, but cytosines remain cytosines if they are methylated, okay? So it has different consequences on the cytosine depending on the methylation level. Well, that's cool, because U's, uracils, are sequenced as T's, because it's a T analog chemically, right? So that means that when you do this conversion with bisulfite, well, you just have to do your next generation DNA sequencing assay now, right? Some of the DNA that was methylated will remain, Cs will remain Cs, so you will sequence them as Cs, but those that were not methylated, they have been transformed in uracils, so they will be sequenced as Ts. So now you have a very easy way to know what is methylated or not. You simply sequence, and you see all the places in the genome where CPGs, CGs remain CGs. It means were methylated, because otherwise they would have been transformed into TGs, into UGs sequenced as TGs, right? So your reads where they're lying in the genome, you just look at CPGs. Are they still CPGs? Yes, it means methylation there. Are they not CPGs and TPGs? No, it means I was not methylated. So simply by bisulfite treating the DNA and do the next generation DNA sequencing, you can have DNA methylation maps at the genome level. Clear enough? All right, so the same way, and why is it important, is that as I told you, DNA methylation, and this is shown in this particular slide as this sort of light gray circles or dark brown circles, the different types of methylation, no methylation, high methylation. This is important because on average, they don't spread randomly across the genome. For example, genes that will be expressed, this is a transcription start site, normally on average, they will be DNA methylation poor, right? So that means that when a promoter is highly methylated, it means that normally those genes will be silenced, right? When there is no methylation, it's a good sign to organize the chromatin and say, you're active, go on the A compartment, if I'm clear, okay? So, and that's also, you know, that some of the CPGs are also placed in the gene bodies, in the nucleosomes that follow the transcription stop site. So this is relatively rich in methylation. And depending on whether there is methylation in the introns and intron boundaries, it will condition the splicing. Right? Genes will be spliced differently depending on where DNA methylation is placed along the intron-exon boundaries, for example. So what I want you to keep in mind is that DNA methylation layers is just one layer, but that layer starts to organize the genome in a particular way so that it gets expressed or not. It tells the CTCF as well, oh, you should come there to make your insulator job. You remember the CTCF that makes the loop that we discussed for separating tags? This is some kind of information that the cell exploits to actually notice, okay? For example, I don't want to be too long here, but if you look at the transcription start site, so this is where a gene will start being transcribed, basically. You see here, I just focus on the blue part, the rest is not important for now. You just count as blue the average methylation level. You realize that there is poor methylation around the TSS, the transcription start site, and then it goes up into the gene body, right, on average. So you realize that along the gene, the DNA methylation levels are not random, right? And they are not random specifically because it's information. It's information that helps the genome to get organized. And it's even more important because if you look at DNA methylation again, that will be those bars that you see at the bottom, depending on whether a gene is high expression level in blue or low expression level in blue, you will have different levels of methylation. As I told you, when expression is high, you will have lower DNA methylation around the TSS, and when expression is low, you will have relatively higher DNA methylation. So you see DNA methylation as a sort of switcher that turns on or off DNA methylation, the expression, gene expression, sorry, right? You see that? You also learn in this particular graph that the way the nucleosomes are placed, that means that the way the chromatin is organized is not random along the gene as well. You see that this is highly rich here in the nucleosome. There is another nucleosome, a third nucleosome, and so forth. So that means that the way nucleosomes are placed is not random as well along the gene, right, along the TSS. And this is because, remember, all this needs information to be placed correctly. So DNA methylation participates to this sort of placement. So I mentioned just now, sorry, I don't know what happened here. I just mentioned DNA methylation, but I also told you that nucleosomes are important because it seems that they are placed in a way that is not random, right? This is not average. There is peaks of where they are located. So we have to have words about how do we even know that, right? Again, because there are methodologies that exist that help us create nucleosome maps as well along the genome, so we know how to annotate the genome for, oh, this is a place where there is a nucleosome that is positioned in this particular cell type, okay? Remember the ENCODE and the FAN consortium. So for example, I'm going to cover two of the many methods that are available to actually gain information about nucleosome placement. One is called micronuclease digestion, MNAs, and the other one is called Hi-C, and that's going to be two important things for you to digest. So what is MNAs? Is that imagine that you have DNA, we have the chromatin, when you extract the DNA, before you do that, you treat the cells with formaldehyde. You know, this thing is something that cross-link molecules together, especially DNA with DNA, DNA with proteins. They will be cross-linked, right? So now they are joined chemically. They are not separate anymore, right? So it joins everything. Then you extract the chromatin, which is all stable because you lock the system using the formaldehyde, right? And then what you use is these micronuclease, MNAs, that allows digestion of the DNA. But when you use that, you realize that the DNA that you will put on the gel is not a smear of the DNA. It's actually a ladder-like shape of the DNA that you have on electrophoresis. And you see that those different bands that you have on the gel, they are multiple of 150 or 170 base pairs. Right? They come in blocks of the same distance, if you want. And this is so because the micronuclease, what it does is that it breaks the DNA in the spaces where there is no nucleosome. Right? So it tells you that the DNA that is wrapped around the nucleosome is 170 base pair long, 190 base pair long. And it can only cleave the DNA in the spaces where there is no histone base. Okay? That's why the result like this. It's 140. Right, and then you can put that on a high-throughput DNA sequencing machine to do this sort of thing. And what it gives you is information about where the nucleosomes are placed. And there are two concepts that I'm going to use later in the ancient DNA course, so bear with me here. So when you have DNA sequencing information after the micronuclease treatment, after the MNAs, you could do two types of analysis really that are very interesting. So imagine that these things are the sequences. So you know that when you sequence, for example, with MGR or with Lumina, you will have oftentimes mates reads, right? So you will have one read and another read by the technology you use. So this is what is presented here. This read has another one on the other direction, this read on the other direction, and so forth, right? So that's why you have pairs like that. They are aligned. Okay, when you have that, you can do a very simple analysis, which is calculate the distance between those reads that are pairs, okay? You see that between that one and that one, the distance is that one, between the second one and the second one, the distance is that one, and so forth, okay? So you could calculate the physical distance between your reads, okay? And you could do that on each and every pair. Once you have that, you could plot, and this is what you see here, the distance plot, that is the distance that you measure here, and the frequency of the distances you find, right? For example, the distance at 50 is found a given time, at 100 is found another time, and so forth. So it's a very simple graph, but you realize that that graph has a periodicity. You see that? So it peaks at 147, 150, which is the size of the distance between two nucleosomes, right? This is on average, you would cleave here and cleave here on both sides of the nucleosome because of the properties of the MNAs. But you also see that you have a 10 base pair periodicity. You see it goes smaller peaks every 10 base pairs. This periodicity is the nucleosome folding. It takes 10 base pairs for the DNA to wrap around the nucleosome. It takes 10 base pairs for a loop of an helix to be folded. Okay, this is what you see here. So we call these sort of graphs the histogram because it's a graph of the distance, okay? We could also do a phasogram, and I'm going to talk to you about this afternoon again, a phasogram, which is slightly the same information but not calculated the same way. What you do here is not the distance between the mate pairs, but between reeds that are aligned in the same orientation. So you calculate the distance between that one and the next one, that one and the next one, that one and the next one, for all the combination in any window of the gene, okay? So you calculate the distance along the same strand of the DNA, right? And if you plot that, it gives you this sort of graph, which is the distance, the phase, we call it, because they are in phase along the same strand, and you count how many you find. You still see that there is peaks every here 190 base pairs. So it's typically the distance between two successive nucleotides. I need to stop in 5 minutes, right? That's correct, sure? For this one, okay. But I need to finish that. That's a very important concept. And so you know that with this assay, you learn a lot about the way things are positioned, where nucleosomes are positioned. Okay? And this is how we can tell that in the TSS, for example, there is a nucleosome-free region here. So when a gene is expressed, normally there are no nucleosomes before the TSS and low methylation level, whereas as you go into the TSS, DNA methylation goes high and nucleosomes get phased very well after the TSS. Okay? I want you to keep in mind that no such layers is independent from the other. They self-inform all of those layers to result in the organization of the genome that I mentioned and also in the gene expression. I told you that there were different types of analysis we can do, and the one that I want definitely to cover now before we take a break is the Hi-C. So that's a different type of assay that tells you a lot of information about the chromatin compaction and how the genome is organized as well. So what the IC is doing is you have the DNA, and what you do is you will actually fix the cells with formaldehyde as you did with the MNAs, so no difference. But instead of using MNAs, which is micronuclease DNAs, you use another type of DNAs, which is a restriction enzyme, RE in my slide here, restriction enzyme. And when you do that, what the restriction does is that it will break the DNA at motifs along the gene, right? Think about any restriction you may have done in the past. Then what you do is that you actually do the digestion, and then you will actually put the biotin and fill in the reads, so to repair the DNA, so that now you could transform that into a DNA library that you could put on your favorite DNA sequencing platform. The reason why you do that, you may have a sort of an idea about that, is that it will actually link through the formaldehyde regions of the genome that are nearby. Because to link things, they need to be close by. Otherwise, you can't be linked with something that is very far away from you. So by doing this thing, it will tell us some information about which regions in the genomes are nearby, are close to each other. Why is it important? I told you, there are compartments. You remember? There are paths. You remember? There are territories. Right? So this methodology is going to tell us where those territories and compartments are. And this is how it looks. So in this particular plot, what we show is a chromosome and the same chromosome we use here in 3 as a restriction enzyme. We could take any enzyme. In that case, that's N3. It could have been any type, RSPA2 or whatever. In that case, that's N3. So what we show here is typically a dot plot. We make a plot with dots, a dot plot. And we put a dot when in the Hi-C information, we have a read that aligns there that has another read located ahead at any place along the chromosome. So when we put the dots here, that means that that part of the chromosome is in contact, with the same part of the chromosome. Not surprising. Me, I'm located with myself, right? So oftentimes you will find me with myself. But what is more interesting is that it tells you that this part of the chromosome also has contacts far away. You see, this part is the beginning. It has contacts in red with this part of the chromosome, which is further away. This part of the chromosome has contact further away in the chromosome, right? You see that? So that means that even though they are not in the same position in the chromosome, they are spatially close by. Otherwise, you wouldn't have ligated them together. You couldn't have cross-linked them, right? So those maps of contacts, they tell you immediately that there are some organizations, spatially of the genome, in which some groups, like for example here, you see this square, there are smaller things that are not necessarily far away, but not at the very same location, that are actually in contact with each other. They are in the same spatial place of the genome. These are short distance. We call it short distance because it's a few megabases, right? But there are some that are much longer distance. You see that? Maybe almost a chromosome harm in it, which is probably 20 megabases apart, okay? So why is it possible? I hope that I was clear that you understand that. Is that this is the sort of summary of what I've just said. It's simply because you have this spatial organization of the genome. You have places where, because of the CCF, it forms loops where long parts of the genomes are actually forming a loop, and so therefore places that are far away are actually close by spatially when they are in the nucleus, okay? So that's why here you will have a contact between something here and something here, even though they are separated by long loops. So the loop will be here in the plot, and this long thing will be the distance that will be covered here, okay? And you have some organizations, such as the TAG, that is at this scale or at that scale. It depends on which TAG you are thinking about. Okay, so really please think about the genome as spatially organized. The last thing that I want to tell you this morning for this lecture is that the same way inside a chromosome there is an organization, there are chromosomes that are more closer with each other, because the map that I've shown is within the same chromosome, but actually you could do the map between chromosomes. So here what you look at is contacts between chromosome 1 and chromosome 2, contact between chromosome 1 and chromosome 17, and so forth. And you put a red as there is more and more contact, and you put a blue when there is a lack of contact, okay? You realize immediately that some chromosomes here, they are closer with each other. They have more contact than they have with anything else, for example. So it tells you that some chromosome in that particular experiment and that cell type, it means that those chromosomes, their compartments are closer with each other, spatially. Otherwise, they wouldn't have such contacts, right? Okay. The very last thing, and then we break, that's correct, is that maybe it's confusing because I gave you a lot of information, but the take home is that the linear genome doesn't exist. It only exists on the screens of your computers because we read that as letters, right? But in life, it's actually organized spatially with domains that will be inactive in the B compartment, some domains that will be active in the A compartment, and all this is shaped in the nucleus simply because there is tons of information available to the cell to allow this organization, such as DNA methylation, such as the way histones are acetylated or methylated or phosphorylated, and the way CTCF are bound to proteins or not with conditions where the loops are formed and organize this sort of super complex organization of the genome. I think I don't need to cover all this because it's already done. I just want to conclude to go back to my original take-home message, which was, why do we care? We care because it's important. That's expression, that's phenotype that's shown you. But why do we care in the context of animal domestication? Well, if you take German foals, which is the ancestor of the chicken, and you do some methylation map for some red jungle foal that will be selected for being afraid of humans or not being afraid of humans, high fear or low fear of humans, you can select for that trait. So you do that for 5 generations. You select the red jungle foal for you are afraid of me or not. So you do selection on this, and then you do DNA methylation map of the 2 groups. And in this particular plot, you look at regions in the genomes that do not have the same DNA methylation levels in the 2 groups. So it will go high when there is a differential methylation in the 2 groups. What I call a difference methylation is a region that is high in methylation in one group and low in methylation in the other group, DNA methylation. Right? Well, you realize that there are parts of the genome where it's significant. So that means that when you select on behavior, your trait is you're afraid or not of your breeder, basically. It doesn't just select for maybe the DNA changes, but it also co-selects, it seems, for differential DNA methylation. So that's why it's very important. Because what you do as a breeder, what we've done during evolution, has actually reshaped the way the genome is methylated, henceforth the way the genome is organized, henceforth the way genes are potentially expressed. Okay? So that's why we really have to care about all these additional factors. I think I will stop with that one for now. We have 15 minutes of the break, and then we will have 2 more this morning, right? And the next 2 ones are going to be about microbes and diseases.